Okay, so now we're on to part 2 of the cell cycle notes. I am at home, so in the background you may hear my husband, Joaquin. You want to say hi? Yeah. Say hello.
Hola.
And my baby who's sleeping. So, and now on to business. Okay, so you should be on learning target number 3. Learning target number 3. I'm pretty sure everyone in class covered this, so this is more of if you were absent. You will need to, those of you that have this will need to fast forward to wherever we left off in class. So, learning target number 3, recognize cells both increase in number and differentiate, becoming specialized in structure and function during and after embryonic development. So, pretty much how do we get from here to here? How do we get from this to that? So first of all, when you talk about cells, we've got somatic cells, we have gametes. These are 2 different types of cells. You need to understand the difference between these. I would make sure in your notes that you go ahead and you highlight somatic and gametes. Those are vocabulary words that you need to know. Okay, so somatic cells, those are body cells. They are diploid. Diploid means you have a double amount of chromosomes. So for example, us, we have 46 chromosomes. That is a diploid number because it has double. You've got chromosomes from mom, chromosomes from dad. That 2N is a symbol that you will see for diploid. So whenever you see 2N, you need to know that that's diploid. Okay, then you also need to know somatic cells divide by mitosis. Alright, then we've got the second type of cell. The second type of cell is called a gamete. Gametes are your egg or sperm cells. Okay, they are haploid, which means they have half the number of chromosomes. So half of 46 is 23. So an egg cell has 23 chromosomes. A sperm cell has 23 chromosomes. They join together in fertilization, and they make a zygote, and then that zygote then is going to become a whole bunch of body cells and go through mitosis. Now, in order to make gametes, we've got to go through meiosis. That's the only time we've got meiosis going on, is to make these gametes. Okay? Alright, so beginning of the human life cycle, we're going to start with gametes. Gametes, once again, those are the egg and the sperm cells. They are going to fuse together, which means they're going to join together, become one. When they join together, they no longer are called gametes because they go from being haploid to diploid in a zygote. Zygote's another vocabulary word that you should know. A zygote is basically a fertilized egg, okay, which then is going to go through G1, S, G2, and mitosis. This is important because this is going to increase the number of cells. This is also going to, once we increase the number of cells, this is going to start, the cells are going to start being specialized and differentiated into things like heart cells and brain cells and limb cells and all those important cells that make up you and me. Okay, this right here, this is the, I guess you could say the progress of a human as we start out. We got a zygote here. That is the sperm and the egg. When they come together, create the zygote, then it's going to go through mitosis. Mitosis is going to increase the number of cells, and then it's going to start differentiating the cells out here. You're going to have, some spinal cords in your brain, in your heart. It starts differentiating them, and then they travel to their separate regions to become the brain, spinal cord, so on and so forth until we get beautiful little babies. Alright, so regulating the number of cells, cell division is going to increase the number of somatic cells. Somatic cells, vocabulary there, that means your body cells. Then mitosis, that's going to be the division of the nucleus. Cytokinesis is division of cytoplasm and everything else. You need to understand the difference between mitosis and cytokinesis. That would be a great test question. Alright, so regulating the number of cells, apoptosis. This is, you saw this in your POGL that you did Monday and Tuesday last week that we graded. Okay, apoptosis, this is programmed cell death. This is going to prevent abnormal cells from proliferating. So right here you can see this guy's got a tail. Something as it was going through the cell cycle, apoptosis did not occur, and this guy ended up with a tail instead. Then we've got, what else is on the screen? Enzyme mediated. That means it requires enzymes for apoptosis to occur. Okay, next. So, learning check how or what changes occur to allow a zygote to transition into fully formed multicellular organism. Take a moment and read through these. Pause the screen. Okay. Correct answer is D. It undergoes mitosis to increase in size, and then the cells are going to specialize for different functions, like brain and heart and liver and kidney and all that fun. Alright, now we're on to, finally, learning target number 4, talking about the different events that occur in the cell cycle. So, cell cycle, this is we're talking about from mitosis all the way back around full cycle to mitosis again. Okay, there are 3 parts. There's interphase, mitosis, cytokinesis. It is driven by enzymes called cyclins. Okay, these enzymes, when you get a buildup of these, this is what allows a cell to progress past the checkpoints. The checkpoints are G1, G2, and M. These are the checkpoints that you saw in your animation you did last week. Okay, once it moves past the checkpoints, okay, then cells are going to be able to divide. So, interphase, this is most of a cell's life. Some cells are permanently stuck in interphase. Okay, you learned about GO from your POCL. Okay, and cell function continues as cells prepare to divide. So as a cell is in this whole portion here, it's continuing as normal. Alright, so we will be doing a lab next week where you'll be looking at cells, and you should be able to see a cell like this with all the stuff in here and know that that is an interphase. Okay, that's what it looks like. So let's talk about the checkpoints. There's 3. There's G1, G2, and M. Once again, we got to have cyclin in order to move past those stages to progress. Okay, and the checkpoints, you're probably going to have to push pause on this so that you can get all of this. Alright, the checkpoints, they're going to govern whether a cell progresses to the next stage. They're like the backup or the double checks. This is the cell as it goes through G1. It's going to check out the DNA, and if it's damaged, it's going to destroy the cell. Okay, if it's not, great, it's going to move through S2. And then it's going to move through G2 again. Once it hits the G2 checkpoint, mitosis is not going to occur if DNA is damaged or if it's not replicated correctly. And then finally, our last checkpoint is M. This is mitosis will stop if chromosomes are not properly aligned. Okay, and as we go through each one of these stages, we'll talk about those checkpoints. So, let's see what else we have here. The default is to stop unless overridden by go-ahead signals. So, this process is going to stop at each one of these checkpoints until it gets the go-ahead. That go-ahead is cyclin, gets enough cyclin there. Intracellular signals detected by control center determine if key processes have been completed. Okay, so interphase. G1 is the first one we're going to talk about over here. This is organelles. They're going to double in number. And G1 is going to accumulate materials needed for division. At the end of G1, we're going to hit this G1 checkpoint. This molecule P53 is going to stop the cycle if DNA is damaged. So why would you not want to pass on damaged DNA? Well, your DNA is your instructions. So if it's damaged, you're not going to get the correct instructions. Okay, yep, that's it. Alright, then we talk about the S phase. S phase here, DNA is going to replicate. That's it. Okay, so we're going to talk a little more about how it replicates. So first we're going to talk about chromosome. Okay, DNA, technically what is a chromosome? Okay, a chromosome is any string of DNA. So this right here, what I've just circled, that is a chromosome. Okay, now it also is considered a chromatid because it is part here of this whole chromosome. So it gets really confusing. And in my class, I want you to know that this X is a chromosome and each half of the X is a chromatid. Okay, what holds them together is what's called a centromere. That's that little part right in the middle. So right there, that would be the centromere. Okay, and then this would be a chromatid and this would be a chromatid. Alright, chromatin, this DNA, okay, it wraps around histones or protein balls. So we'll get a little more into that in a moment. Alright, so DNA, or the histone complex, nucleosome, supercoils, and as it supercoils, it creates chromosomes. So what does it mean? It means it goes like that. And then supercoil again, and you get these long stringy things that look kind of like X's. So a chromosome is a condensed rod-shaped DNA molecule during division. Alright, so here we've got the DNA, the helix. Take note of the size there. 2 nanometers. Super, super tiny. The DNA helix, I'm sure you guys have learned this before. You've got your 2 parts right there to the DNA strand. Okay, think about this. Bunch of Christmas lights. If it's not organized, it's this huge, disgusting mass that's just impossible to get straight. Okay, so what happens in our cells is that our DNA strands are going to wrap around what are called histones. A histone, that's what these little blue balls here are. Okay, so if you've ever wrapped Christmas lights, if you wrap it around your arm, you can help it not be as tangled. Okay, so the very first step is that our double-stranded DNA is going to go through and wrap itself around these histones. Okay, notice size. We're getting a little bit bigger here, from 2 to 11 nanometers. Alright, then all of those histones with the DNA wrapped around it are going to start wrapping around each other. Notice our size, 30 nanometers. Then all of the DNA that's wrapped around the histones and the histones that have wrapped around each other are going to wrap around each other even more. Notice the size, until finally we get this nice condensed chromosome right here. DNA, we started at 2 nanometers. Okay, we condense it to 1400 nanometers. So it gets much larger, which is why you can see these shapes underneath the microscope. Okay, so G2, this is the synthesis of proteins needed for division. Okay, so we're in this last part of interphase. It double checks growth and replication. And then we hit the checkpoint, G2 checkpoint. It's going to stop the cycle if DNA is not done replicating, so if it needs a little more time, we're going to stop. Or if it's damaged, it's going to stop it. Once again, we don't want to pass on damaged DNA to the rest of our cells. Okay, so learning check. What would happen to the daughter cells if the S phase of the cell cycle was removed? Okay, apparently you didn't put the rest of it. So, think about it. What would happen to the daughter cells if the S phase of the cell cycle was removed? We'd have no DNA in one cell. Or, you'd have half the amount that you need. Okay, and this would not survive, this would not survive. So the S phase is really crucial for a cell to survive. Okay, next 5, explain how cell cycle is controlled and what occurs when control of cell cycle is lost. I think we'll stop there for tonight. Alright, so have fun, be good with the substitute, do not talk, do your work, do your foldables, check the website for the next set of notes, okay? See you later.